Hello everyone, welcome to Cell Biology Part 2. So in this particular class also we are going to discuss easy to moderate level questions. In the next part we will discuss about moderate to difficult level. So I will solve nearly 250 questions very shortly from this cell biology topic. So that's why I want every one of you to compulsory make use of easy questions also, moderate questions as well difficult questions also. So focus on the three types of questions compulsory without fail. Okay, now listen. Let's quickly understand. Mitochondria are called as the powerhouse of the cell because we all people commonly know, right? We started studying from the 11th standard. Mitochondria will produce abundant amount of the ATP because in the inner membrane of the mitochondria, several electron acceptor complexes are present which are involved in the synthesis of ATP. And they never release the oxygen. They use the carbon dioxide, no, and mitochondria not at all involved in the synthesis of any proteins directly or indirectly. Mitochondria can synthesize proteins, that is true. 10% of proteins it can synthesize, but because of the synthesis of protein, we never call it as the powerhouse of the cell. We call it as the powerhouse of the cell because it is involved in the synthesis of ATP, not to get confused with that. Then the option, question number 2, pick the wrong statement about the mitochondria. Option 1 says that mitochondria divided by the fission. Absolutely true. Mitochondria always shows the binary fission. They possess linear DNA molecule. I told to all of you many number of times, whatever the DNA present in the mitochondria is this circular mitochondria. Sorry, circular DNA, which is the supercoiled. Is it? Not to get confused with that, but they said that it possesses the linear DNA molecule. This is the wrong statement. They produce energy, right? Even though mitochondria is present in the eukaryotic organism, friends, they contain 70s ribosomes only. This is also right statement. Hence, the correct answer is option 2. The cell organelle which has DNA, not to get confused. Endoplasmic reticulum, which is involved in the synthesis of protein, do not have DNA. Golgi complex, which is involved in the sorting, do not have DNA. Lysosome contain only hydrolytic enzymes. It do not contain DNA. Mitochondria is the organelle which contain the DNA. Question number 4. The non-functioning of this cell organelle inhibit the cytokinesis. This is also very good question, friends. Ribosomes required for protein synthesis. Mitochondria required for energy. Chloroplast is required for photosynthesis. So which is involved in the cytokinesis, which is the Golgi complex. Not to get confused with that, because Golgi complex is the one which is doing the lot of protein sorting. If the Golgi complex is not functioned, some of the most important proteins like tubulin proteins, which are involved in the formation of the spindle fiber, which is required for the nuclear division. At the same time, according to the given question, cytokinesis, a cytoplasmic division. So in the cytoplasmic division also, there are certain proteins are required. That proteins are getting modified or processed or sorting in the Golgi complex. So that's why if the Golgi complex is not functional, cytokinesis never occur. Next question. One of these is a cell organelle common in both Monera also as well as the Protista also, which is the common, friends, which is the ribosome. Is it? Mitochondria, chloroplast, it is not present in the bacteria. Of course, lysosome also, you know, right? It do not contain lysosome, but it contain lysozyme, which is an enzyme. Not to get confused with that. Next one, the inner compartment of the powerhouse of the cell. So you have to understand the question. Inner compartment of the powerhouse of the cell. It doesn't mean that they are asking about the inner membrane. That is wrong statement. This is outer. This is the inner. The middle space is the inter. These two are membranes. They're asking inner compartment. Not to get confused with that, friends. So the inner compartment will be what? Matrix. Number of chloroplasts in a cell of chlamydomonas will be, of course, friends, in most of the organisms, I mean to say, I'm talking about the chlamydomonas, the number of chloroplast is only 1, not to get confused with that. Double membrane bound cell organelles of the cytoplasm are. So we know very well Nucleus is a double membrane. Chloroplast is the double membrane. Mitochondria is also double membrane. But the question says that of cytoplasm. Not they did not ask about the nucleus, which is the chloroplast, as well as the mitochondria. Anyway, they did not given the option of nucleus also. Now, not to get confused with this particular question number 8. Why answer is 3? Why not answer is 4? That's also a very good question you have to understand. Why you did not consider the nucleus? Why I have considered only the chloroplast and mitochondria? What is the reason? Because friends, nucleus is entirely different, because it is the nuclear genome, and these two are what? These two are the extracellular genomes. Is it? Mitochondria and chloroplast we can call what? Extra chromosomal inheritance. Is it? They are not following the Mendel pattern also. They will always follow the non-Mendelian pattern. So that's why you should not include the chloroplast and mitochondria with the nucleus. Always remember that. Hence the correct answer is option 3. Chloroplast and mitochondria. Not to get confused. The question 9 says that, what is the question number 9? The question number 9 says that pigments required for light reactions of the photosynthesis are present in pigments. What are the pigments? Chlorophylls are the pigments, carotenoids are the pigments, xanthophylls are the pigments. Where they are actually present? They are present on the membrane of the thylakoids, right? Obviously direct question. Ribosomes contain, most of the students will get confused that ribosome means, friends, they will think that there is only presence of the RNA. All the students remember for the exam, ribosome contain RNA also, protein also. You remember, right? We studied about the not one protein, friends. There are many proteins are present in the ribosome. If you look into the whether it is a prokaryotic ribosome or whether eukaryotic ribosome, one of the major protein or an enzyme present in the ribosome is what? Peptidyl transferase enzyme, which we also called as a catalytic RNA, which we also call it as the ribozyme. Right. So ribosomes are the combination of RNA plus protein. It do not contain any kind of a DNA. Hence the correct answer is the 4. Now, question number 11. Mechanical support to the cell is provided by. So, most of the students, when they heard the name mechanical support, they will directly choose the answer of only cell wall. No, friends. Cytoskeleton also plays a crucial role in the mechanical support. This is actually very important, cytoskeleton. You know, right? Cytoskeleton only contain a lot of proteins. Cytoskeleton contain tubulins. Cytoskeleton contain actin filaments. Cytoskeleton contain intermediate filaments. They are required for the structural support. They are required for the motility also. They will perform multiple functions. Among those, one of the major function is the mechanical support. So what my point? So mechanical support of the cell is provided by cell wall also, as well as the cytoskeleton also. Not to get confused with that point. Then the question number 12 says that cytoskeleton is absent in. Undoubtedly, eukaryotic cells are actually contain the cytoskeleton. Prokaryotic cells do not have any modified cytoskeleton also. So what will be the answer? Bacteria do not contain the cytoskeleton. We all people commonly know that. Mycoplasma or PPLO do not contain it. Blue-green algae also do not contain the cytoskeleton. Not to get confused, these 3 do not contain cytoskeleton. Please remember not to get confused with that. You have to remember here. Flagella of the prokaryotes lack what kind of a configuration? When we are reading about the flagella, friends, we will read it. So it contain 9+2 arrangement, 9+2 tubulins arrangement in the axoneme, which you can't observe in the prokaryotes. Hence the correct answer is 1. And in where this particular 9+2 will be arranged within the center of the flagellum or within the center of the cilia, that is only they have asked as a separate question. The central core of cilium or the flagellum is known as, what do you call it as? You call it as the axoneme. Never ever get confused with that point. Next, question number 15 says that one centrosome contains dash centriole. It's a common, right? One centrosome contains what? 2 number of centrioles. Very direct question. Which of the following plastids are helpful in the storage of the starch? You know very well, chromoplast, it is not at all involved in the storage, it is involved in the synthesis. Why? Because friends, it is having a pigments. Amyloplast is the one which is involved in the storage of the starch, and hence the correct answer is the number 2. Chloroplast is also involved in the synthetic function. Proteoplast is involved in the storage of the proteins. So what will be the correct answer? The correct answer is number 2. Then question number 17, 70s type of ribosomes are found in. This is also simple question, makes you confused. Every prokaryotic cell contains 70s ribosome, but we already studied many number of times. According to the endosymbiotic theory, even chloroplasts and mitochondria, even though they belong to the eukaryotic organism, they possess the chloroplast, they possess the mitochondria compulsory. Hence, what will be the correct answer? Option 2. Question number 18. Which of the following substances are stored? This is also very, very good question, friends. Elyrioplast. This is very important, friends. Which of the following substances are stored in the elyrioplast? I want you to remember this is the protein. Right. So actually, friends, remember my words. This elyrioplast can be break down by several enzymes. All those enzymes can be activated by the hormone gibberellic acid, which are actually required for the process of seed germination. Whenever I am explaining you the hormone gibberellic acid, we will discuss over there. Next, question number 19. Smallest cell organelles, which is called as protein factory. We all people commonly know that. Lysosome contain hydrolytic enzymes. Vacuoles, they are involved in the hemostasis and storage. Endoplasmic reticulum involved in the synthesis of protein. But you have to understand what is the question they asked. The question they asked is smallest cell organelle, which is called protein factory. You have to understand, if I ask you a question, are you studded with ribosomes? So that was not given in the option. So then you should not confuse. You should not choose the answer as endoplasmic reticulum. At least if they give in the option R here, then you have to think about it. They just mentioned endoplasmic reticulum, so you no need to choose this option. The correct answer will be obviously ribosome only. And question number 20, the ribosomes are made up of. I already told you, ribosomes do not contain any kind of a DNA molecule. Ribosomes only contain the RNA and protein. Hence the correct answer is 2. Cilia and flagella both have. What is the common thing? Just now I told you, right? Cilia and flagella, in the central part, what they are having? They are having axoneme, a wheel-like structure, which is having what kind of arrangement? 9+2 microtubule arrangement. When we are discussing the class, you will automatically understand. And it is not required for any protection, only present in the protozoa. Wrong statement. And it is not at all the outgrowth structure, it is the inner structure, it is the central part of the cilia or flagella. Hence the correct answer is the number 1. Now listen everyone, which of the following pair, very good question, lack the unit membrane? That's a very good question. Nucleus and ER contain the membrane, you know that. Mitochondria and chloroplast contain the membranes. Golgi body and lysosome, they contain membrane, but extremely thin membranes. So the point is, friends, they contain the membrane, whether it's a thin membrane or thick membrane, doesn't matter, they contain membrane. Hence, fourth option is also right. Ribosomes and nucleolus. Right, the nucleolus, you know, right, where the rRNA is getting synthesized, where the protein gets synthesized. Ribosomes are also where proteins are getting synthesized. They do not have any kind of a membranous structures. Hence, the correct answer is option 3. Not to get confused with that. Within the cells, ribosomes are found in. RER do not contain, microbodies do not contain, chromoplasts do not contain, Golgi complex do not contain, lysosomes do not contain. Only the 2 organelles that contain the ribosomes among the given options are chloroplast and mitochondria. Not to get confused with that. Next, question number 24. Glycosylation of lipids and proteins occur in the cell organelle. I have told you very clearly to all of you, there are 2 types of glycosylation will happen. Glycosylation means what, friends? Addition of the sugars. And we know that there is a O-linked glycosylation occur, and friends, there will be a N-linked glycosylation occur. I have told you very clearly, N-linked glycosylation occur in the RER lumen, rough endoplasmic reticulum lumen. And friends, where does the O-linked glycosylation taking place? Golgi complex. Anyway, in the given option, they did not mention anything about the RER. Hence, what will be the correct answer? Undoubtedly, Golgi complex. Suppose example, in the exam, if they mentioned both Golgi complex and peroxisome, all the students, sorry, in the Golgi complex and mitochondria, Golgi complex and rough endoplasmic reticulum, every student, remember, they will compulsory ask in the question whether it is a O-linked glycosylation or is it a N-linked glycosylation. Compulsory they will give that particular question, not to get confused with that. Hence, what will be the correct answer? Undoubtedly, the correct answer will be the number 1. Question number 25. Which of the following cell organelle is just posed to the nucleus and contain cisternae? This is also a very good question. I want every one of you to understand the question correctly. Which of the following cell organelle is just posed to the nucleus and contained cisternae? See, most of the students will get confused. This is outer membrane of the nucleus. This is the inner membrane of the nucleus. Is it? The outer membrane of the nucleus is connected to what? It is connected to the rough endoplasmic reticulum. We all people commonly know that. Now, in the given option, rough endoplasmic reticulum is not there. Undoubtedly, the rough endoplasmic reticulum will be connected to what? It is connected to the Golgi complex only. Is it or not? So, lysosome, from the Golgi complex, proteins will go to lysosome. Mitochondrial peroxisome is completely irrelevant. What is the correct answer? Which is the Golgi apparatus. And you know, right? RER and Golgi apparatus, RER, SCR, Golgi apparatus only contain the cisternae. Any of these 3 organelles do not contain cisternae. There also you can easily explain the answer. Hence, the question number is 4. Answer is 4, I mean to say. Question number 26. Type of RNA found in the matrix of the mitochondria. I told to all of you friends, you can find all type of RNAs. mRNA, rRNA, tRNA. Why? Because mitochondria is involved in the synthesis of their own proteins. If they want to synthesize the protein, friends, it is compulsory. There is a requirement of all the type of RNAs. It is mandatory. Is it? Next, the term suicidal bag is applicable to cell organelle. Today, in today's class, we are going to discuss very briefly about the lysosome. Right, lysosomes are said to be the suicidal bag. We will discuss why and what are the type of lysosomes, like a primary lysosome, secondary lysosome, phagolysosome, autolysosome. We will understand the what is heterophagy, what is autophagy, what is autocytolysis. It's a very important one. Okay. So the answer is lysosome, which is a straightforward question. Question number 28. Smooth endoplasmic reticulum is well developed in the cell which synthesizes. You remember, I told you in which organelle smooth endoplasmic reticulum percentage is more when compared to all other cells. Always remember, friends, liver. Liver is the organ in which smooth endoplasmic reticulum, compose or comprise most of the part after the mitochondria. In the liver, mitochondria and SCR will occupy the major percentage. First mitochondria, then SCR. And we all people know that SCR not only involved in the synthesis of lipid. This is a direct question. But every student remember for the exam, SCR also plays a very crucial role in the process of detoxification. Never ever get confused with that particular point. Then question number 29. The Golgi complex plays a major role. All the students remember, I'm telling you right now, Golgi complex will helpful in the sorting. Second thing, and the most important one, I told you, right, the Golgi complex plays a key role in the glycosylation. These two are the 2 fundamental functions performed by the Golgi complex. So option 1 says that as energy transducing organelle. Wrong statement. It is in digesting proteins and carbohydrates. It is not at all involved in any digestion. In trapping the light. Why the Golgi complex will trap the light? It do not have any kind of a pigment. So it is involved in the addition of sugars to the lipids and proteins. By that, lipid converted into glycolipid, protein converted into glycoprotein. Very simple point. Hence, what is the correct answer? The correct answer is the 4. Not to get confused. Question number 30. When will green tomatoes turns red? See friends, first of all, you all people has to remember one very important point. What is that? Tomatoes red in color because of a specific pigment. We all people know that. What is the name of that particular specific pigment? The name of the pigment is called lycopene. Is it? Lycopene is the specific pigment which is reflecting the red light. So chloroplast contain chlorophyll. They only reflect the green color. So when the chloroplasts are converted into chromoplast, then the lycopene reflect the red color. So answer will be what? Option 2. Carbon assimilation is the irrelevant option. Respiration is irrelevant option. New chloroplasts are never made. That's a wrong statement. Don't think that new chloroplasts are developing. Chloroplast only modified or converted into chromoplast, which contain a specific pigment, reflect the red light. By the tomatoes turns red. The name of the pigment is lycopene. Red chili also contain lycopene only, not to get confused with that. Most of the hydrolytic enzymes of the lysosome function set. Today also we are going to study. Friends, if lysosome contain, all the students remember my words without confusion. Lysosomes contain more than 150 hydrolytic enzymes. More than 150. And friends, if all these enzymes wants to work, they require a suitable pH of 3 to 5, which is what, friends? Acidic pH. So you have to understand very interesting point. If you take the lysosome enzymes and if you keep it in the neutral pH or a basic pH, they never ever be active. If they wants to be active, always they require acidic pH. Hence the correct answer is number 4. Question number 32. If we separate the cell organelles of a living cell, which part should be alive? This is also very good question, friends. ER will degrade fast, cell wall will degrade fast, ribosomes also will degrade fast. So if you separate the cell organelles, chloroplast will be alive. Why? Because chloroplasts contain the pigments. They are trapping the sunlight. You know, right? Sunlight is the major source of energy for each and every cell and each and every organelle. Correct, na? So ER, cell wall, ribosome, they never trap any kind of energy actually. So that's why chloroplast will be alive, not to get confused with that. Hence the correct answer is number 2. 33. Which of the following cell organelles is considered to be in catabolic enzymes? Just now I have explained it to you, right? Lysosomes contain more than 150 hydrolytic enzymes. You know, example, we will discuss lipases, which break down the lipids. Glycosidases, which break down the glycosidic bond, nothing but sugars. Proteases, which break down the peptide bond, nothing but proteins. So friends, what are these? They are all breaking down enzymes. That is what we can call catabolic enzymes. So catabolic enzymes only present in the lysosome. Golgi body do not contain any enzyme. It contain enzyme, but they are involved in addition of sugars. Mitochondria, no. Endoplasmic reticulum, which is the irrelevant. It is the one which contain more than 150 different type of hydrolytic enzymes which are involved in the breakdown, which are catabolic. Answer is 1. The endoplasmic reticulum, that is studded with ribosomes. We all people commonly know that ribosomes, if they are present, they are obviously involved in the synthesis of protein. This kind of questions are very important, friends. You people will forget. Remember, highest number of the mitochondria are found in, I want you to remember this compulsory, meristematic tissues. Now the question is very clear. Why not parenchyma? Why not collenchyma? Why not sieve tube? You know very well, meristematic tissues are the fast growing tissues in the plant. Is it or not? Of course, yes. They are the one which are fast growing tissues. So friends, you have to understand, whichever are growing fast, all the students listen my words, whichever are growing fast, they obviously require abundant amount of energy. So if they want abundant amount of energy for its growth and development, obviously what they require? Mitochondria. Hence the correct answer is option 3. See the question number 36. Mitochondria and chloroplast are concerned with the transfer of cellular energy. Is this statement is correct? Now you only tell me. Chloroplast never involved in the production of ATP. See, don't get confused. Please don't consider with respect to the photosynthesis. Then you can ask me a question. Sir, actually, in the chloroplast, in the photosynthesis, chlorophyll pigments are involved in the production of ATP. Then why you are saying it is wrong? No. They're asking which are concerned with the transfer of cellular energy. So transfer of cellular energy is mediated only by the mitochondria. So option 1 is wrong. Option 2, so both are not involved. So it is no, only mitochondria, but not the chloroplast. Hence the correct option will be undoubtedly 2. Option 4 they say that no chloroplasts are, but not mitochondria. This is wrong, right? Only mitochondria involved in the transfer of cellular energy, but not the chloroplast. Hence the correct answer is 2. Question number 37. The biomolecule synthesis by the rough endoplasmic reticulum are modified here being released. This is also very, very good question, right? Suppose, friends, in the rough endoplasmic reticulum, what will synthesize? Tell me, proteins. Now they're asking question, proteins, where they will reach? They will reach to their targeted area. Yes or no? They will reach to their targeted area. Before they reach their targeted area, they have to undergo the modification compulsory. The question asked where they will undergo the modification. That is the question. Where they will undergo the modification. Friends, the modification always occur in the Golgi complex. I told you, right? It is the one which is acting like a sorting station. Not to get confused. Correct answer is 1. And there is no role of vacuole, lysosome, peroxisome with respect to the protein synthesis. These 3 are irrelevant. Question number 38. After packing materials, Golgi operators secretes then to the, very important, right? See, once all the materials or the proteins or the enzymes, once they got packed into the Golgi apparatus, it is going to secrete into different compartments. You cannot conclude that it is only secretes means, friends, release. You cannot conclude that it is only releasing into the inside of the cell. You cannot conclude that it is releasing only into the outside of the cell. No, you cannot say that. So it can be released into the all other, inside also as well as the outside also. Hence the answer will be both 1 and 2, not to get confused. Question number 39. The pigment which is not found in the chloroplast is, they're asking which is not found in the chloroplast. Carotenes are present in the chloroplast, you know, right? Antenna pigment. Chlorophyll, reaction center. Xanthophyll also antenna pigment. Anthocyanins are not found in the chloroplast. This is the answer. Question number 40. Membrane-bound vesicular structures formed by the process of packaging in the Golgi apparatus is. What the question they are asking? In the RER, every student has to remember proteins get synthesized. That proteins are modified where? In the Golgi apparatus. And that proteins finally packed in the form of what? Vesicles. Correct, na? And that vesicles, what do we call that particular vesicles? That vesicles only we can call lysosome enzymes. That vesicle contain enzymes of the lysosome. Now that vesicle go and fuse with the lysosome and they release all the enzymes into the lysosome. In the lesson you automatically understand in the today's class only. So hence the correct answer is obviously 3. Question number 41. Which of the following statement is correct? Ribosomes do not contain DNA. Of course, this is true statement, right? Ribosomes do not contain any kind of a DNA. It contains only what? RNA and protein. Nothing else is present in the ribosomes. Eukaryotic ATS ribosomes break into 50S and 30S. It is wrong statement, right? ATS ribosome break down into 60S and 40S. So this is wrong statement. Plasmodesmata are found as an intracellular junction. Wrong. Plasmodesmata is present between the two cells. It is never ever connect intracellularly. It is connecting two cells in between the two cells. Ribosomes were discovered by Beadle and Tatum. So option only 1 is right. Option 2, 3, 4 are wrong. Hence the correct answer is 1. Right. Question number 42. A cell biologist used an ultrasonic method to disrupt the cell and found fragments in a cell and found fragments in cell organelle, which served as a site of ATP synthesis. The moment you have to understand ATP synthesis, undoubtedly you can answer the question as mitochondria. What I want you students to remember in this particular question, which technique is used? This is the question I want every student has to remember. Which technique is used to disrupt the cell layers or the cell membranes of the mitochondria? That is important for the exam. So which is that? Ultrasonic method. That is also you can expect in the form of a question. Ultrasonic method is used to disrupt the cell and separate the fragments. Correct answer is number 1. One of these is an important function of vacuole. Most of the students, when they are reading the vacuole, they just think that vacuole is involved in the storage. Today class only we are going to discuss. Every student remember, vacuole ultimate function is homeostasis, maintenance of internal environment that is crucial for every living cell, which we called osmoregulation. Among the homeostasis, one of the processes is osmoregulation. There are many homeostasis processes are there. Among many homeostasis process, one of the major one is the osmoregulation. It is not involved in any synthesis. It never involved in the energy production. It is never involved in the breakdown. Only storage and only homeostasis. Hence the correct answer is the 1. Select the false statement. Ribosomes and centrioles are the non-membrane bound organelle. Of course, true. Centrioles do not have membrane. Ribosomes do not have membrane. This is right. Ribosomes are enveloped by double membrane. This is wrong, right? Plasmodesmate are the cytoplasmic connection between the cells. Absolutely true. Cell membrane is semi-permeable or selectively permeable. That is also right. So, sorry. What's the question they asked? Ribosomes and centrioles are non-membrane bound organelle. This is right statement. And what is the question they ask? Ribosomes are enveloped by double membrane. This is false statement. Option 3 is also right statement. Option 4 also right statement. Correct. They asked false statement. So false statement will be the option 2. Ribosomes are not enveloped by any membranes, but they said that it is double membrane. So it is the wrong option. Question number 45. Which one of the following pairs is not correctly matched? They are asking correctly matched. Nucleus, which contains genetic information, which is the DNA. Right. Cell membrane is selectively permeable. Golgi complex involved in the secretion. Absolutely true. Leucoplasts, they said the synthesis. What I told you, leucoplasts do not contain pigment only. If do not contain pigment, how it is involved in the synthesis? It is only meant for storage. So what will be the answer? Undoubtedly the answer will be the 4. Under what conditions do lysosomes cause autolysis? In this today's class, we are going to show this. We are going to explain this very elaboratively. All the students remember, under what condition lysosomes shows autolysis, which means that it is not going to show the autolysis every time. Only during the condition of starvation. Only during the condition of starvation, it will break down. By that, that enzymes will break down. The enzymes will break down a particular biomolecules which are involved in the production of energy during the starvation condition. And it is only possible under starvation. Autolysis is not a generalized process. Autolysis is a specialized process which will occur at a specific condition called starvation. If excess food is there, why we have to break down? It is not a method of injection, not a method of respiration. It is a direct method of starvation. And in the last class, I told you, how can we classify the plastids? Pigments and function. These two are the two major parameters that we used in order to classify the pigments. Classify the plastids. Type of plastids present in a stem tuber of Solanum tuberosum. What's the point you have to remember? So, stem tuber. Tuber is what? What is the function of tuber? It is majorly involved in the storage. It is not at all involved in any synthesis. Since it is involved in the storage, undoubtedly leucoplast, because chloroplast and chromoplast, these two are involved in the synthesis function. because they contain pigments. Hence, the correct answer is leucoplast. The pigment molecule of a chloroplast are associated with the proteins of, I already told you, right? On the thylakoid membrane. On the thylakoid membrane, electron acceptors are present. Photosystem 1 is present. Photosystem 2 is present. ATP synthase enzyme is also present because they're all present on the LHCs. Hence, the correct answer is 1. The following organelle protect the cell from toxic effect of H2O. In the last class also I told you. In the peroxisome, there is an enzyme called catalase. Catalase is the one which is actually breaking down the hydrogen peroxide, not to get confused with that point. Hence, what will be the correct answer? Option C, peroxisome. So friends, in this particular class, what we studied is the EG to a moderate level. So in the next classes, we are going to study moderate to difficult level, right? Okay. That's all for now. I'll see you in the next class. Thank you.